Hello, welcome everyone. I want to thank you all for joining us today and also thank the members of our Patient Outreach Committee for planning out this educational series, many of whom you will hear from as they share their expertise on these topics. We're excited to have you here for this first Lunch and Learn of the series and hope you will tune in each month as the topics are meant to build off one another. This is an extension of the ASGCT Patient Education Program, where our website currently hosts tons of reliable resources that educate on gene and cell therapy and are free to share. As we head into today's talks, Jennifer Helfer will be our moderator. She joins us as the head of patient advocacy and engagement of Encoded Therapeutics, a gene therapy biotech company focused on pediatric CNS diseases. She has a strong background in the pharmaceutical industry, patient advocate, nonprofit, and bench research, and is passionate about engaging patient communities to improve therapeutic development.
Thank you, Allie. Hello, everyone. Thank you for joining us live or watching on demand. We're really excited to be here kicking off this Lunch and Learn series with two amazing presentations, one by Dr. Philip Tai and the other by Dr. Rachel Bailey. Throughout their presentations, please submit any questions you have in the chat, and we'll do our best to get to as many of them as possible later in today's webinar. First up, we have Dr. Philip Tai, who is an assistant professor at the University of Massachusetts Chen Medical School and Department of Microbiology and Physiological Systems. Dr. Tai is also the director of Vector Innovation Discovery and Engineering Program. His current research focus is on the development of next-gen sequencing methodologies to profile AAV vectors, the discovery and characterization of natural AAV capsid isolates for vectorization in gene therapy, and the development of safe and efficacious vectors. So with that, I thank Dr. Tai for joining us, and I will hand it over to you to walk us through the basics of gene therapy.
Great. Thank you for the introduction. It's my pleasure to kick off this meeting. And so I think the goal for my set of presentation slides, because I'm the first speaker, is to really give an introduction into gene therapy for those that may have just heard about these magnificent platforms, or you may just be a patient family that just heard about these technologies and are thinking about the possibilities for these wonderful platforms. And so in order to understand gene therapy, I think we have to first understand genes, right? So as you may know, genes are what make us biologically unique, right? So they're made up of DNA, which is, of course, the blueprints to build enzymes or proteins that perform various crucial bodily functions. Unfortunately, our genes can gain mutations, and this can come in the form of as our cells age, or we are exposed to certain chemicals, or they can actually be inherited. So any small changes can actually be very damaging, right? So small changes within the genes or even surrounding the genes may have large impact on cellular function, such as breathing, walking, or digesting food. And so if we have a normal gene, we may produce normal proteins and function normally, but if we have a mutated gene, we may produce abnormal proteins, or we may even have a lack of that protein entirely. And so in order for us to really understand human diseases, we really need to have a good understanding of these diseases that may be affected on a genetic scale. And so in terms of inheritance of genetic diseases, they really come in these three main forms. They come as autosomal recessive. So this is obtaining a mutant gene from both parents, which will cause disease. Autosomal dominant mutations or diseases where you receive a mutant copy from one parent, which will cause a disease. And then there are also a class of genetic mutations that are X-linked, where you inherit them from your mother, that will also cause disease. And so what is then gene therapy? So gene therapy is simply put, is an introduction, removal, or change of genetic material within the patient's cells. And this normally comes in the form of a transfer of genetic material into the cells of a patient to repair a gene or compensate for a loss of a gene to treat a specific disease. These concepts aren't new. They've been around for a fair amount of time. So this is best illustrated by this Time magazine cover hailing gene therapy as the future of medicines. And so the overall action of these gene therapy platforms is the introduction of this genetic material and so into cells. And so once this gets into the cells, the agent can correct the faulty gene by reducing the levels of a disease-causing protein, increasing the production of a disease-fighting protein, or producing new or modified proteins entirely. And so one of the major benefits of these gene therapy platforms is the advantages in discovery and development over traditional means of developing drugs. And so typically in the classical means of drug discovery, labs may be screening tens of thousands of compounds at a time in order to come up with a shorter list that can be tested in animal models, either small animal models or large animal models at preclinical stages. And then once we find some promising candidates, these can then be moved into human clinical trials. And this can be a very laborious line of investigation because it requires several years of study as well as the involvement of thousands of volunteers. And then all of this investigation is then reviewed by the FDA, and then this can potentially be moved into the marketplace. Because of the overall length of these type of studies, the cost to carry out these investigations are very costly. However, for gene therapy, because most of the platforms that are utilized to deliver therapeutic gene products have been tested for many years, many of these investigations can be abbreviated. And because gene therapy is really targeting for rare diseases and diseases that are defined by single gene mutations, this process can be very much abbreviated. And because the FDA now recognizes that these rare diseases have limited populations for volunteers, they have combined, multi-phase trials into single phase so that these can be abbreviated, the investigation be reviewed by the FDA and then moved into the marketplace. So how does gene therapy work? Gene therapy can be broken into four main categories. The first is gene replacement, where you replace a non-working gene with a healthy one. These are to target mainly the loss of function genetic diseases. And then there are gene therapies that promote gene silencing. So this is the activation of a mutated gene that can become toxic to cells. And these are related to gain of function genetic diseases. And then there are gene addition strategies, which is the expression of exogenous gene products to impact the disease state. And then lastly, gene editing, where you generate a permanent manipulation of a gene in a patient's genome. And so at the heart of gene therapy really is the delivery mechanism. And so typically the genetic material is transferred into the cellular target with a vector, right? And the vector is simply just a vehicle that carries the therapeutic gene product, or the therapeutic gene, sorry. And the most promising vectors currently are derived from viruses because they've evolved to enter cells very, very efficiently. The positive aspect of some of these viral gene therapies is that you can remove the viral genes that can potentially be pathogenic, and these are entirely replaced by the engineered genes. And so in theory, the viral vectors are non-pathogenic and will not cause toxicity. And so once these get into the cells, the genes will make functional protein and target the disease-causing faulty gene. But there are actually two main methodologies to execute gene therapy. Those are broken down to in vivo and ex vivo strategies. And so in vivo is essentially the direct delivery of these vectors into the patient in order to deliver the therapeutic gene product. However, in ex vivo strategies, one can take out patient cells, culture them in the lab, and then subject them to the therapeutic vector in order to manipulate or engineer the genome. Then these are further passaged and cultured in the laboratory and then delivered back into the patient in order to achieve treatment. And so in terms of gene delivery vehicles, there are actually several types. They can be mainly broken down into two types, the viral vectors, as I had mentioned before, and then non-viral vectors. And so non-viral vectors actually span just the delivery of naked DNA, which was used in the early 60s in the first few trials for gene therapy. But viral vectors can also be complex with lipid particles or liposomes. Now there are new platforms that are referred to as lipid nanoparticles that are used to deliver genetic material for therapeutics. But for the viral vectors, these can be further subclassified into RNA viruses as well as DNA viruses. And as you can see here, these viral vectors come in many different shapes and sizes, and they can be utilized for different purposes depending on the therapeutic target as well as the size of the gene therapy, therapeutic of vector genome, and whether you want the therapy to be persistent in the cell that it's targeted or transient. And actually, what's amazing is the fact that many of us have already experienced exposure to these viral vectors because they came in the form of the COVID-19 vaccines, right? So J&J and the AstraZeneca vaccines utilize adenovirus vectors, whereas the Moderna and Pfizer vaccines utilize the lipid nanoparticles to deliver RNA payloads. And although these were used at relatively low doses, these vaccines really represent the largest use of therapeutic vectors to deliver gene products, and these have given us a positive outlook for these therapeutics to cure and target other diseases. And so within the landscape, the three main viral vectors that are in use currently are those that are based on adenoviruses, which make up about 50% of the viral vectors that are being tested in the clinic. And then about 22% of these are related to lentiviruses. And then the rest, 28%, are based on adenovirus vectors. And this is actually the vector platform that our lab focuses on. And it's really an up-and-comer in terms of a viral vector of choice. And so in terms of the vector design, these are conceptually very straightforward. Here, of course, is using AAV as an example. The two main viral components that are essential are the viral capsid protein. So this is the protein coat that packages the vector gene. And then essential viral elements. And so within AAV, these are inverted terminal repeat sequences that flank the therapeutic gene cassette that are essential for production as well as persistence in the cell. And then the genetic component that's delivered can be simplified into blocks of genetic sequence that serve specific purposes. So here I'm illustrating protein coding, transgene that's being expressed here in this green block that can be driven by genetic sequence that promotes the expression of these transgenes called promoters. And then there are other blocks of genetic sequence that either promote transcription or stability, like the polyadenylation sequences. In addition, you can also deliver non-coding RNAs. And so these may be related to platforms that promote gene editing or gene knockdown therapy. And these can be driven by other classes of promoter sequences that drive non-coding RNA sequences. And in addition, in order to improve safety of the viral vector, you can also engineer tissue-specific enhancer and promoter constructs. So the idea being that if you deliver these viral vectors systemically, either by intravenous injection or injection into areas that may potentially leak into other regions, you want to control this type of expression, and you can use these specific cassettes in order to drive tissue-specific expression. And so I don't want to go too in-depth into the multitude of vector designs. I encourage everyone to take use of the patient education resources that are available on the ASGCT website. And those go into detail about the different kind of vectors and vector strategies that are being employed. But in terms of gene replacement strategies, There are several, two that are based on AAV, which has gained a lot of attention in recent years. One called Luxturna, which is a gene therapy used to treat patients with inherited retinal disease caused by a mutation in RPE65 gene, and Zolgensma, which is a gene therapy to treat spinal muscular atrophy, or SMA, where patients have a loss of this SM1 gene, which causes a degeneration of motor neurons. And so there are more platforms that are on the rise. And these encompass, of course, also gene knockdown strategies. So gene knockdown is a term that considers strategies that can diminish the amount of native transcripts that are generated. So these strategies may target, for example, Huntington's disease or a spinocerebellar ataxia, cancer, or even viral infections, where expression of toxic genes may cause disease. And so if we deliver cassettes that drive the expression of microRNAs, which are a natural mechanism to break down messenger RNAs in all of our cells, or siRNAs to promote destruction of RNAs. These are a viable means of gene knockdown for disease-causing genes. And then lastly, there are these classes of constructs or designs that promote gene editing. And these, within recent time, has caught a lot of attention because it is really a means of tailoring these genetic strategies in order to target diseases in a very precise way, right? In a permanent manner. So gene editing disrupts or corrects faulty gene elements, DNA elements within a gene. It uses highly precise system to make changes within the host cell genome. Effectively, it uses an enzyme that can cut the DNA at a location that's defined by a guide RNA sequence. Either you can create mutations within, directed mutations within the disease-causing gene, within the DNA itself, or you can insert a functional sequence within the DNA in order to reverse the mutation. And the most well-known are these CRISPR-Cas9 platforms that have been developed in recent times. So I just wanted to show a slide to basically illustrate the necessary components. One is this Cas9 enzyme that can be targeted to a specific region of interest based on this guide RNA sequence that can also be designed into gene therapy vectors to target the editing event to a specific region in the genome. This causes a double-strand break within the gene, which can either promote non-homologous end joining in order to create either insertions or deletions to disrupt the gene product, or it can undergo a homology-directed repair with a donor template in order to precisely correct this sequence. And investigators now have been developing new additional platforms based on this Cas9 platform to promote specific base editing as well as editing of RNA. And the idea to edit RNA is to provide a much safer means of correcting mutated genes so that you're not targeting the DNA itself. And so to close, I wanted to finish up on this slide in terms of trying to understand gene therapy and how we can tailor it, right? So the important questions that we must ask are, what is the target, right? So every different gene has different challenges. Whether the problematic gene is a secreted gene versus whether it's a cell-specific gene, these are important questions to ask. The other is, what are the host barriers, right? And so this will may come up in our question and answer session, but things like the host immune system still poses a major challenge for gene therapy application. The third bullet is, of course, what's the best vector to use, right? And I alluded this, and I couldn't go into very much detail, but these are very critical questions, right? What vectors, whether they be viral, whether they be lipid nanoparticle based, are the best to use to tackle the problem. And what's the best route of delivery? And this goes along the same lines of what the best vector is, right? So where do you target the gene therapy? Is it a directed muscle injection, or is it a systemic delivery, or is it intracranial injection in order to target cells of the CNS? And then what are the manufacturing bottlenecks? And I think Dr. Bailey may talk about some of these aspects, because one major bottleneck for these therapies is the manufacturing process. They still require a lot of resources, and they're still based on the production within cells. And this is a very costly strategy to employ to create these drugs. And then finally, really, what are the risks, right? And I hope that as a group that we can start talking about the risks. And so I look forward to the discussion, and that will be the end of my slides. Thank you so much.
Thank you so much, Dr. Tai. That was super helpful overview, and I really loved the use of your animations throughout. I kind of got distracted by watching some of them. So we have received quite a few questions in the chat, but we'll hold those for later, and we'll go over to Dr. Bailey next for her presentation. Reminder to those watching live, please do submit any questions you have in the chat, and we'll get to those right after Dr. Bailey's presentation. Dr. Bailey will be presenting on the current landscape of gene therapies. Dr. Rachel Bailey is an assistant professor in the Center for Alzheimer's and Neurodegenerative Diseases and the Department of Pediatrics at the University of Texas Southwest Medical Center. Dr. Bailey's research focus is on the development of gene therapies for neurological disorders and facilitating the translation of these potential treatments for use in human clinical trials. With that, I turn it over to Dr. Bailey.
All right, well, thank you, Dr. Helfer, so much for the introduction, as well as thank you to Dr. Tai for giving the overview of gene therapy. There may be a little bit of overlap within my presentation, but that way we cover everything that you'll need to know. So as a reminder, this is the first of our Lunch and Learn series. So today I'll be discussing the current landscape of gene therapy. And so I'll be reviewing the process for developing a gene therapy from bench to bedside. And then I'm going to highlight gene therapies that are currently approved and those that are in the development pipeline. And then the diseases that are being targeted in these applications. And so finally, I just want to provide a highlight of some of the benefits and challenges to the field. And really, the goal of this session is to provide an overview of the field, as we will be covering many of these topics actually in much greater detail in our future Lunch and Learn series. So I hope you all are able to attend those as well. So let's start with the clinical process for developing a first-in-human gene therapy drug. So Dr. Tai talked about this somewhat, but I'm going to go into a little greater detail on each of these elements. So as was mentioned, the preclinical study happens before a clinical trial can start. The treatment is tested in cell models or animal models, and then these studies check if the treatment will work and how much of it is needed to see a positive effect, so determining the dose. It also tests the safety by looking for any negative effects. And then depending on the availability of cellular and animal models, as well as the complexity of the disease that is to be treated, the length of time needed to complete these studies can actually vary greatly from disease to disease and often can take several years. If the preclinical results are promising, though, the researcher then designs the study for humans. So as part of this, I've termed it as trial readiness, where it's not necessarily part of the normal timeline, but I think it's important because as researchers prepare a study for humans, the next step would then be to submit a pre-investigational new drug application or an IND application, a pre-IND application to the U.S. Food and Drug Administration, or the FDA. And within that document, it would detail the preclinical results to date, the plans for formal toxicology testing, drug manufacturing, and then the tentative clinical trial protocol. The FDA then reviews this document and provides feedback on any changes that would be needed prior to approving an IND document and starting a clinical trial. So with this feedback, researchers can then complete studies that are necessary to meet that feedback, and they can move forward with manufacturing the drug as it needs to be made to be human grade, as opposed to the preclinical studies, which can be a research grade. And with making a drug for humans, the quality assurance is actually quite high, because before you can put this in a human, it has to be documented and detailed exactly how it was made, what is packaged, and that it's made to the quality that is appropriate for use in humans. And the other thing I want to point out that can be done during this time frame is a natural history study, potentially, if it's needed. So for some rare diseases where there's a low number of affected patients, it may be necessary to conduct a natural history study to understand how the disease naturally progresses in the absence of a gene therapy treatment. And this helps the researchers to identify outcome measurements that can then be used within a clinical trial to determine how well the drug is working. So once the preclinical and trial readiness steps are completed, then it can move forward into the application period. So this is where, as I mentioned before, the formal IND application can be submitted to the FDA. The FDA then strictly reviews how the treatment is made, the preclinical data, and then the clinical trial design. And it is their job to make sure the researcher has done everything possible to minimize risk to the study volunteers. So once the FDA agrees the risk to volunteers are minimized, there are other regulatory approvals that need to be gained. And so those usually come in the form of an institutional review board or an IRB and the Institutional Biosafety Committee, which are found at the site for where the clinical trial is to be started. So once all this regulatory documents are approved, then they can move forward into a clinical trial. So as Dr. Tai mentioned, the clinical trials are conducted in phases. So every phase is modified to help answer a different question to better understand the treatment. So as always, the safety is a priority in each of the phases. If a study is not deemed safe, it will not move into the next phase. So as part of these studies, it includes monitoring for adverse events, which are any unexpected health problems that can range from minor to serious. The researchers also collect information on whether the events are related to the treatment, the severity of them, the frequency of them, and then how they can be resolved. And then in turn, these become the expected side effects to be aware of for that drug. So for the individual phases, phase 1 tests if the treatment is safe for a small number of patients. Sometimes it can also be determined, it can be used to determine the dose to be given. Phase 2 expands the number of patients receiving the therapy and determines if the responses to the therapy are favorable or useful while still making sure it's safe. And then in phase 3, this is typically the longest and has the most individuals enrolled in it. So in this phase, the main goal is to prove that the treatment gives the desired result while being safe in an even larger group of patients. So as Dr. Tai mentioned, some of these phases can be combined to make the evaluation process more efficient. So this is typically done for serious and rare diseases for which there is a clear unmet medical need, meaning it's a disease whose treatment or diagnosis is not addressed adequately by the therapies that are currently available. So this really puts an emphasis on the need for this gene therapy. So finally, if the results of the final phase of the clinical trial are showing that the treatment is safe and effective, a biological license application or BLA can then be submitted to the FDA. Following thorough review, the FDA makes a final decision about whether the treatment will be approved for use. So this is when a treatment becomes FDA approved and then is made available to the public. So something that is important to note here is that between the preclinical study and clinical trials, as I've mentioned, this process can take many years to conclude. Clinical trials alone sometimes take 8 years or more. So there are many factors that can go into the duration, including study planning, authorization to run a trial, ethics review, funding, research materials such as patient information, consent forms, and then the monitoring systems. However, the FDA has provided various expedited pathways to accelerate the process for these therapies that meet certain criteria while still maintaining safety. And so part of that can be, whereas we discussed, combining phases. Another way to potentially accelerate this process could be in the early period during the preclinical studies and trial readiness, rather than to do some of those processes in parallel, that they are done together, rather than doing them sequentially, that you do them in parallel. So for example, the human drug could be manufactured prior to completing the preclinical safety and efficacy studies and before receiving regulatory feedback. However, this does come with a high level of risk regarding resources and finances, so careful consideration would need to be taken before going down that path. All right, so having discussed what the process is to develop a gene therapy drug, let's now discuss where we are in those processes. So as part of the goal to develop gene and cell therapy resources, ASGCT has partnered with Informa Pharm Intelligence to generate quarterly data reports that cover the landscape of gene, cell, and RNA therapies. These reports are available online through the ASGC website. And so in the next several slides, I will be showing results from those reports, and which were generated in January of 2022. So here I'm showing the globally approved therapies. For the next series of slides for what I'll be discussing, the gene therapies are being divided into 3 separate categories. One is gene therapy drugs, which includes drugs that deliver DNA to the host cells, and that can include genetically modified cell therapies. There's also broken down into RNA therapies, which are drugs where pieces of RNA are delivered to cells. So that can include antisense oligonucleotides or ASOs, as well as the messenger RNA therapies, such as the Moderna and Pfizer COVID vaccines. And then there's also then the non-genetically modified cell therapy. So those are 3 categories we'll be looking at. So as we can see from this report, over half of the globally approved therapies, so these are therapies that are approved by different regulatory agencies across the world, and not necessarily all within the US, only a few of these are within the US. We see that the majority of them, 62%, are the non-genetically modified cell therapies, while 21% are gene therapies and 17% are the RNA therapies. Here, though, I would like to highlight 2 FDA-approved AAV gene therapies. So these are gene therapies that are approved within the United States. The first is Luxturna, which is a gene replacement therapy for Leber congenital amaurosis, where patients with mutations in the RPE65 gene experience childhood blindness. So in 2017, this drug became the first approved AAV drug therapy in the United States, and also which became the second in the world. And then came Zolgensma, which was approved in 2019 for the treatment of spinal muscular atrophy. So children with this rare genetic disease suffer from debilitating and often fatal muscle weakness. A one-time injection of Zolgensma, though, into the blood of children that were less than 2 years of age was shown to significantly improve muscle movement and function, quality of life, and importantly, survival. So the effectiveness of this treatment highlighted the significant impact that a gene therapy can have for patients where no other treatment option is available for them. And so this drug in particular greatly increased the interest of pharma researchers and patients in driving forward gene therapies in general. So really this field has just exploded over the last 3 years with the interest in moving gene therapies forward. Mm-hmm. So what I showed previously were approved drugs. now what I'm showing are the drugs that are in development. So that is going to be a much higher amount. So we have 89 approved gene therapies globally. However, currently there's over 3,000 therapies that are within the preclinical to pre-registration phase of development. And so here we see the majority of those are actually gene therapy. So that is the modality that is currently being worked on the most by researchers to drive forward to treatment to patients, with then RNA therapies and cell therapies being about 21 and 23% respectively. The other thing I wanted to highlight, which Dr. Tai spoke of also, is that of the gene therapy, so out of that green pie, 89% of those therapies that are in development use viral vectors for delivery. So some of those treatments do not use viral vectors, but the majority of them do. And as Dr. Tai alluded to, adeno-associated virus, or AAV, has really risen to the top of the viral vectors that are most frequently used. And so from the report that was gathered by ASGCT and Informa, you can see here the breakdown by different viral vectors as to how many are currently being used in gene therapy development. And then to touch on the actual pipeline. So this is looking across a year span of time for how drugs are changing, or how many drugs are in preclinical, phase 1, phase 2, phase 3, and then pre-registration, and how this is changing over a year of time. Overall, the majority of them are in the preclinical phase, which is to be expected. However, we do see the greatest growth seen in the phase 3 development in terms of the number of drugs that increased in this category when we moved over a year span. Of course, the increases in the number of preclinical drugs continues to develop. And although there were no new gene therapy approvals in Q4, we see that the pre-registration values were fairly even from October to January of this year. Interestingly, though, in 2019, the former commissioner of the FDA, as well as the CBER director, Peter Marks, had made a joint statement where they predicted that the FDA would be approving 10 to 20 cell and gene therapy products each year by 2025. And so although we have not quite reached that goal yet, I think it's important to keep in mind there has been a pandemic during that. So I would say I'm actually still very encouraged by the growth we continue to see in the gene therapy pipeline, despite the fact that we're through a pandemic. But it's still thought that we'll continue along, potentially along this amount of approval, although it's likely to be on the lower side. So the other thing I wanted to touch on is what are the most commonly targeted therapeutic areas? So again, from these graphs, what I'd like you to take away is that when we look at the ones that are in preclinical through pre-registration phase, we see that most of the drugs are actually anti-cancer drugs that are being produced. But then right behind that is actually drugs for rare diseases. So this is going to be largely monogenetic diseases. And so that's not surprising, where if you have a mutation in one gene and you know what the cause is, that using gene therapy to then replace that gene or to affect its expression in some way is makes it a viable target for gene therapy. And then we see neurological products are the next in line after that. If you look at the amount that are actually in the clinic being tested currently, again, the majority barely is rare diseases, and then the other half is the cancer products. So what I showed previously was all types of therapies. Here again is a focus on just AAV gene therapies. So here is a figure from a review done by Kuzmin et al., where they focus on the clinical landscape of specifically AAV gene therapies. And so what I'd like you to take away from this is that of the completed trials that were available at the time of this report, what they found was that gene therapies really focused on targeting 4 tissues, which was eye, liver, muscles, and then the CNS, which was then reflected also in the top 10 most indications that were pursued. So this is where a lot of the research efforts are going currently in the clinical trial. So where are we going? I hope you've gained from what we've talked about so far that we have RNA therapies or oligonucleotide therapies that have been approved, and that includes Spinraza, also for SMA. We have CAR T cell therapies that have been approved, and this falls into that gene therapy bucket. And we also have AAV transgene delivery that's approved in the form of Luxturna and Zolgensma. This one in particular, the AAV gene therapy, is again they are drugs for monogenetic disorders, which are going to be a little more straightforward conceptually in terms of you have a mutation in a gene, so you replace a functional copy, therefore you should help the gene. So we're starting to move beyond that in the complexity of what we're addressing in the clinic to actually include gene editing, where now clinical trials are starting with using that modality, as well as moving into the use of engineered iPSC cells. And then this brings then on the horizon where the science is going, which would be the goal of trying to do base and prime editing and really making more personalized therapies, as well as then thinking about, can we do epigenome editing as a way to then try to treat more common diseases? And that would ultimately be our goal, is not only do we have precision medicine, but that we can also develop medicines that can affect more common diseases. So Dr. Tai had touched on some of these as well as to what are some of the challenges that we're facing in gene therapy. And of course, number one in my mind would be safety and immunity. So this is taken into account prior to a patient being enrolled into a clinical trial. Are they potentially excluded from a trial because they may have pre-immunity against a vector that was going to be delivered? There are concerns about immune responses that can be triggered by the delivered drug, whether it be from the vector or if it's from the gene itself that's being delivered. And then as part of the natural course of where we have moved to in clinical trials with delivering gene therapy, part of the normal regimen has become immune suppression of patients. So when a patient becomes enrolled in a clinical trial and they're going to receive a gene therapy, they often undergo a heavy regimen of immune suppression so that when they are given the drug, they don't have a strong innate immune response against the drug. But being on that type of immune suppression can then make patients more susceptible to infections or other diseases they can encounter in their environment that is completely separate from the drug. So we're actually going to be discussing some of these immune concerns at our next Lunch and Learn, so I'm going to leave it there. The other that Dr. Tai did a very nice job of mentioning was manufacturing, is the cost that's associated with it. The demand, part of that is being, from what I showed, with the number of therapies that are in development, there is actually a massive strain on the manufacturing capability to be able to produce the amount of virus that's needed for so many indications, both for clinical trials, as well as considering once you get to approved drugs, how is it sustainable to keep making that? So that's something we'll actually be addressing in, I think, our April Lunch and Learn series, and have experts in the manufacturing world to discuss them. And then finally, other challenges do include the long-term effects. Gene therapy is still relatively new, and particularly as we change our viral vectors or we change how we're modifying genes, it's uncertain what potential long-term side effects there can be, as well as there's questions out as to how long the therapeutic benefit can last. Will this last the lifetime of the patient? Where that is really probably going to be cell type specific. So if you have a cell that rapidly divides and you did not use a vector that integrates into the genome, then the effect could be lost over time. But all this is what still is being learned as we go from trial to trial. And then finally, just thinking about our rare diseases, there are limited populations for those. So it can be very challenging to get to an FDA-approved drug, one from a financial standpoint of getting the funding necessary to go through all those stages to regulatory approval, as well as having enough patients to enroll in each phase of the clinical trial, or even as a compressed clinical trial. And so that is something the NIH is trying to work on as well as different pharma companies are also becoming specialized in rare diseases. So we may get help in that aspect. So then finally, some of the benefits of gene therapy. I think Dr. Tai mentioned these as well, is really it's precision medicine that targets the cause of the disease. And for diseases where they're rare and debilitating, this can be critical. With gene therapy, there is the potential for a single administration, particularly where you use a DNA-based drug, where then the cells will have the DNA to make more of the drug they may need. It has the potential to eliminate or reduce the need for other treatments. And it also is potentially overcoming one of the barriers for neurological conditions, which is the blood-brain barrier, which is typically difficult to overcome with small molecules in terms of them being able to get into the brain, which viruses are quite good now at getting into there. So there's a lot of potential on the horizon for these drugs. So finally, I want to just provide resources for patients and caregivers that if you would like to look up any of this on your own, please check these websites out. And thank you so much for your attention. And sorry I ran a little bit late, but Dr. Helfer, I'll hand it back over to you.
Wonderful. Thank you so much, Dr. Bailey. That was an amazing presentation. Tons of questions have started coming in. I know we're not going to get to them all, so apologies to everybody who's been submitting, but we'll do our best to get through this. Dr. Taya, asked you to put your video on and make sure you're with us so we can go ahead and start these questions. I think the first question that started coming in when we started was around, will the slides be available? The recordings will be made available for this webinar, so definitely check back on the ASGCT website for that, and I believe also the YouTube channel. Second question, we've had quite a few questions come in around IND enabling studies and what type of preclinical studies are needed around both the vectors themselves and the safety of them, as well as other safety measures before we start testing these gene therapies in clinical trials. So maybe Dr. Bailey, I can start with you for that, and then maybe Dr. Tai will go to you.
Okay, sure. So from the preclinical aspect, usually that starts by having a model of the disease of some sort. So this can be potentially a cell model where you have patient cells, or more often it is an animal model, usually a small animal model, that has aspects of the human disease that you can look at to look for efficacy. So you take your virus and you deliver it into this model, and then you look for efficacy markers. So one, you typically look at biodistribution of your drug if it's a whole organism. So where is the drug going? What tissues is it expressing in? You look for your actual transgene expression. So is it expressing, is it functional? And you have to show proof of that. And also, is it working? What benefit is it providing? So are you able to make your animal model live longer? Are you able to fix a biomarker that can be found in the blood? Can you fix part of the behavioral phenotype? To really put together an argument that there is a potential benefit for patients. And that, because with any clinical trial that's a first in human drug, it is a risk, because you don't know until you test it exactly what will happen. So you need to provide evidence to the FDA that there is a reasonable expectation for benefit to a patient. And so along with that then is safety studies. So these are typically done in disease animals, so we call them wild type animals. And as a preclinical researcher, you can do them just in a wild type mouse. And what's important for that is thinking about long-term following. So to deliver your virus into an animal and then to assess that animal over a long period of time, say a year post-injection. It also needs to be done in what's called GLP toxicology studies, so good lab practice studies, or all the acronyms get mixed in there. And that's where it's typically done by a contract research organization that performs that study for you in a larger animal model. So that can be potentially a rat study or a non-human primate study, or even a combination of that. And then the contract research organization will report out how was it tolerated, what was the biodistribution, was there any concerns? And so that's from just the data preclinical side, I guess. And then you get into the CMC, so the manufacturing of the drug. And that's a whole, I don't know if you want to talk. I mean, I'm sorry, Phil, I feel like I've been talking for a while, but if you want to jump in there, or I can keep going. And so again, that's where you have to be able to explain exactly how this drug is being made, how it is being purified. And the FDA has come out with guidelines or state what needs to be met in order for them to say you can move forward with the clinical trial. And part of that also is showing that the drug that is being made for humans is very similar to the drug that was used for your toxicology and preclinical studies. And if there's a big mismatch there, then sometimes they do request that you repeat some of the preclinical studies with that human drug that's being made in a different manner. And that's called a bridging study, where again, you have to show the drug would be the same, therefore you'd expect the same safety and the same benefit before you move into a human. And then there's the clinical trial plan. But I don't think we need to get into that, because I think the question was about data. So. All right, and Phil, if you have anything to add, please do.
No, I just one thing in terms of the CMC side of things. Yeah, we have even been doing studies where we're using next-gen sequencing methods in order to gain better understanding for what vectors can be contaminated with. So, it's been shown, and people know in the field for a long time, that when you make these viral vectors, they're not 100% perfect, right? So there's always going to be some, truncated genomes or contaminating DNAs that come from the plasmid and the host cells that are used to manufacture them. And so there's an acceptable amount of these contaminating DNAs that can go into the viruses. But now what the FDA has started to ask is, are there any potential viral genes that aren't associated with the production process that could end up in your prep, right? So I think this was related to another one of the questions that were typed up, is the presence of replication competent viruses, as well as advantageous viruses that can end up in preps. And so yeah, there's a lot involved in terms of understanding what goes into the prep. Yeah.
Great. Thank you both so much. That was a perfect answer. In a follow-up, somebody else asked about the regulations for gene therapies, and are they the same between the FDA and the EMA and other regulatory bodies?
So from my experience, there are slight differences there, particularly with CMC. There has been. I think there are some efforts to try to bring them to be the same standard more globally, but traditionally between like EU, Health Canada, and the United States, there may be some differences in what's required for CMC. And there may be some differences for what is required in some of the toxicology studies in terms of what type of animal model you use. For example, if you can use a rat versus a non-human primate, how long you need to monitor those animals for. Can you do 3 months, 6 months? Does it need to be a year? So there can be some differences between the regulatory agencies around stuff like that, and particularly CMC. But I feel from the recent meetings I've been in, it feels like they are trying to bring things up to be to the same level globally.
Great. That'd be great if there is that synergy across the agencies. So we're going to walk a little bit away from the IND and kind of that enabling piece. And we've gotten quite a few questions about vectors themselves. And I think, maybe we'll start with Dr. Tai. You mentioned the package. And there was a question around, are there any limitations depending on the different delivery mechanisms and the package they can contain, and what might those be?
Mm-hmm. Yeah, that packaging is certainly a concern in terms of limitation, right? So for example, the AAV vectors that our lab works on has a packaging limitation of about 5 kb. And so when you consider the transgene as well as the regulatory elements that go into it, everything has to fit within a 5 kb size. And so that puts a lot of these, targets out of reach, because, if a gene is 10 kb in size, right, then we have to use other viral vectors, right? So antiviruses have a larger packaging capacity. Even these lentiviruses do as well, but they also have their own, considerations to take in. And so, yeah, definitely those are the limitations.
Great, thank you. Another question came in. This actually came in during the registration, and I want to make sure we get to it. Someone asked, can you explain neutralizing antibodies and what this means for people who are considering treatment with a gene therapy?
Yeah, I can start. So neutralizing antibodies is really an outcome of the immune system recognizing components of either the vector or the transgene itself, right? And so, for example, again, I'm sorry to bring up AAV, it's just the thing that I work with. About 50 to 80% of the human population has seen AAV, right? Because you can consider it almost kind of like a common cold virus, right? Because it's just so prevalent in the human population. And so if you've seen a specific serotype, then you may be limited in receiving this gene therapy. And so this is why a lot of the field has been trying to discover new capsids, right? Capsids that are non-related to human AAVs, or modify capsids that can either detarget the immune system, or they can escape human antigens, right? So neutralizing antibodies. And so these are the type of strategies that are being deployed in order to overcome neutralizing antibodies that may be present in the patient. And yeah, that's a very good question.
And so I would just also add to that. That was great, Dr. Tai, but just to add on to it. So in thinking about clinical trials, typically, a decision is made whether to include or exclude patients that are seropositive, is what they call it, for neutralization antibodies against the vector you plan to deliver. So if you're planning on injecting into the blood, which is where neutralization antibodies are typically located in high numbers, then those patients wouldn't be allowed to participate, because if a viral gene vector is delivered into their blood, the neutralization antibodies would recognize it and then destroy it. So then they wouldn't get benefit from that drug, and then potentially they could have a much stronger immune response. An alternative, which has been shown in clinical trial, is that if you deliver into the cerebral spinal fluid, there are trials where they have injected patients that were positive for neutralization antibodies in their blood, but they could then deliver the drug into the cerebral spinal fluid, and that was tolerated well and it was safe. And also patients still had cells that took up the virus and they were still able to express. So there's more research going on in that area. And then part of it also is, as I'd mentioned, with part of the regimen with then being injected, is going under steroids and other regimens that help regulate any potential immune response you have.
Thank you both. That was super helpful. So we've had quite a few other questions come in around viral vectors and other type of delivery mechanisms. And I just want to remind everybody that our March Lunch and Learn webinar is going to be a deeper dive into viral vectors. So make sure you register and attend that. I know we're getting close to time, so maybe we'll move on and this might be our last question. We'll see if maybe we can squeeze another one in. But maybe Dr. Bailey, we'll start with you. What do you think are the most important considerations of gene therapy that one might need to consider or think about when they're thinking about going under gene therapy for a treatment?
So for a potential patient considering enrolling it, you really need to consider benefit versus risk. If you have a very severe disease where there's no other treatment options, and this is potentially something that shows it could be efficacious, then it may be more favorable for you. There's almost an equation where you have to think about it. Also, as part of clinical trials, there are exclusion criteria, which means even though a patient may want to participate in a trial, they may not be able to if they have some underlying factor about them that then excludes them from the trial. Because it's also about safety. It needs to be safe for a patient to participate in it. And if there are some diseases where there's competition in that arena, and there are actually multiple therapies that are available for one disease, a patient should try to talk to their doctor, look into each clinical trial to see which one could potentially be a better choice for them. Because once you enroll in one AAV vector gene therapy, you're unlikely going to be allowed to enroll in a second. There can be some limitations on how many clinical trials you can participate in. So that should be discussed as well.
Yeah, I was just going to mention a little bit about that, because there is certain psychology in whether you want to enroll in a clinical trial, especially if it's related to a non-life threatening one, right? Because you want to get the best therapy that there's out there. And so if you know that 2 years, maybe 3 years down the line, there's going to be a better trial available, then why take that risk to enroll in a trial when it may exclude you from the next round of gene therapies? And so, yeah, I guess this is something that you should talk to your doctor with, and also get advice from your family and loved ones in order to better understand what you're enrolling into. But I would say though that volunteers that do enroll in these studies, they're absolutely critical, right? In order to help improve the therapies. And so, yeah, that's a great question.
Yeah, it's a risk-reward ratio usually that you have to figure out. And I'd also put a plug in for the ASGCT education website, that there are several diseases where it's been spelled out for what is available for those and considerations for each disease. So patients can look at that as well to see if there's a clinical trial for that, and go to clinicaltrials.gov also to see if they could find a trial for them.
Perfect. And you just answered another question that had come in, so thank you. Unfortunately, I do think we are at time. So I just want to say a huge thank you to our amazing presenters, to everyone who was able to join us live today, and for all your great questions that came in. Also, thank you to ASGCT and NORD for holding this Lunch and Learn webinar series. We're really excited. This is the first of many. They're going to be held throughout the year. And with that, I'll turn it over to Allie to wrap us up.
Thank you all. I hope everyone enjoyed today's presentations. Please know we'll be posting this recorded session on our website early next week. You'll get an email with a link to that. All registrants will. So please feel free to share it. Also, we'll have a short 5-question evaluation at the end. So if you can take a moment to share your feedback, we'd greatly appreciate it. And then registration is now open for that next Lunch and Learn. So as I said, these will be taking place monthly. Come join us on March 31st for an introduction to viral vectors. We hope to see you there. Thanks, everyone. Have a great day.